So Isabelle Eisenmann is a physicist by formation, engineer and physicist by formation. She did her PhD in ENS Paris. And she's now at the University of Amsterdam for a postdoc. And so she studies the dynamics of photosynthetic organism, and in particular Chlamydomonas renartii, which we know well in the lab. So thank you, Isabelle, again, and the stage is yours.
Hello, so thanks a lot for the invitation. And I'm super happy to tell you about some of the things that I did during my PhD in Paris, and some of them in collaboration with Aliénor. So it's a very multidisciplinary work. So I'm a physicist, I'm doing experiments, but we worked in close collaborations with theoreticians and chemists and biologists. And I'm going to tell you about multiple collective phenomena in a phototactic microalgae that you know well, I think, which is Chlamydomonas renartii. So just a quick reminder. So Chlamydomonas renartii is a unicellular microalgae. So it's a single cell. It's 10 micron diameter. It swims quite fast, like approximately 10 times its diameter per second. And thanks to its two undulating flagella that you can see here. And it's also a photosynthetic organism. But as physicists, we see it mainly as an active particle, so which has its own autonomous motion, and which is very interesting because we can control it by light. And this is possible because the single cell has an eye. Actually, it has two arms and one eye, but no brain. And this eye is called the eye spot. It's like a primitive light-sensitive organelle, which is located at the cell periphery, at the cell equator, and which allows it to measure where the light is coming from. So the cell is able to either follow, swim in the light direction, which is called phototaxis. And in the dark, as you can see in this movie, the cells just have a random motion, but as soon as the light is switched on, they all have a very directed motion. So this is real time. And so this phototaxis is very different from bacterial chemotaxis that you may know. So it's not a biased random walk. It's really like a directed motion in a fixed direction, which makes it very interesting for us because we can control it very well. But of course, as a photosynthetic organism, it also has a much more complex relationship with light. So actually, light is not always good for this kind of organisms because too much light can generate photodamage. So if the rate of light absorption is greater than the rate of photosynthesis, so there is some excess light that can be detrimental to the cell. So the cells have evolved a wide diversity of strategies to mitigate, to both optimize light absorption and mitigate photodamage. So they have lots of molecular mechanisms, but maybe the easiest strategy is just to move and go to go toward the light when you need light and to escape the light when you have too much light. So these cells are able to do both positive phototaxis, which is going toward the light, and negative phototaxis, which means swimming in the direction opposite to light. And now, what happens when you put a lot of these cells together? So they can interact in many ways. And today I'm going to tell you about phenomena that are due to shading, two types of interactions. So first, shading interactions, and then hydrodynamic interactions. So shading interactions are due to the fact that these cells, as I told you, are both highly sensitive to light. So the eye spot is extremely sensitive, and they are also they absorb a lot of light. They need to do it for photosynthesis, so they are green, they are full of pigments, so they strongly modify their environment. And so if you have a cluster of cells, it generates a shadow, and this can affect the motion of the cells that are next to it. And for hydrodynamic interactions, well, as you saw in the first movie, each cell swims. So it also modifies its closed environment, which can then advect a cell which is moving next to it. And then when you put millions of such cells together, you can have a lot of complex effects that I studied during my PhD. Okay, so the first collective phenomenon that I studied is purely due to shading interactions. And the setup is very simple. So what we did is simply put 2 milliliters of a cell suspension inside a small Petri dish of 3 centimeter diameter, where the cells are swimming. They are just swimming in their liquid medium, and then we surround it with 16 white LEDs. And in the experimental conditions here, the cells only do negative phototaxis. So in the dark, at first, what you have, this is the top view, you have a cell suspension of roughly a few million cells that are all swimming, and they are all negatively phototaxis, so we can expect them to all aggregate at the center as soon as we switch the light on. But actually, this is what happens when you switch the light on. So at first, it's homogeneous because they just swim randomly. But very quickly, after approximately 2 minutes, you can see this very nice pattern that appears. So here, the dense phase is the dark one. So all the cells condense in these very beautiful branches. And then the branches slowly retract toward the center. And at the end, after approximately 10 minutes, you end up with a very dense blob of cells at the center. So here you can see it again. And clearly, I will show it later, but there is a well-defined wavelength that is selected in this system, which give rise to this very irregular branching pattern. And maybe you can see in the movie that during the branch retraction, you can see that the thin branches, they retract much faster than the wide ones. So they tend to flow and disappear very quickly compared to the big ones. And actually, this tells us about what is happening here. Because if you plot the speed of retraction, so of each tip, of the tip of each branch, as a function of the width of the branch, you can see that, as we saw in the movie, the wider the branch, the slower the retract. And this trend matches an opposite trend in the cell density. So the wider the branches, the denser they are. So the darker they are in the movie. And this is actually due to light absorption. Because if you take a very wide branch, like the one here at the left in this movie, so it's made of lots of cells. So here you can see the individual cells. And because the cells are so full of pigment, the typical absorption length that it's possible to compute it's super small. It's only a few, it's like in the order of 10 micrometers. So if you, so after a few cell layers, you almost absorb all the light, which means that in a very wide branch, in the bulk, the cells almost don't perceive light anymore. And so their motion is just the same motion as they have in the dark, which is just purely random, and here dominated by collision because it's so dense. And only the cells that are located at the interface are able to see the light, and thus tend to swim away from it. So in the very wide branches, the retraction only happens at the interface. So it's quite slow compared to the size of the branch. And on the contrary, if you take a thin one, like here at the right, all the cells inside are able to see the light. And thus, all the cells have a directed motion away from the light. And so you have this net motion, which is way faster than the motion in the wide branches. So this first analysis tells us that it's super important to take that light absorption by the cells coupled to phototaxis is super important to understand what's happening here. And what is actually the mechanism for the branch formation? Okay, so here, please look at the left. So it's a movie, microscope movie of the branch destabilization, of the instability. And so you can see that, okay, at first, so they just swim toward the center, but very quickly you have this branch formation. It's clearly a wavelength which is selected with the distance between two branches. And then you have all of these more complex merging events, and then the slower retraction of the cells. And here we can also say a lot of things actually about the physics of the phenomenon. Because if you take such a microscope movie, and you plot both the cell density and the cell speed, you get this kind of plot. So here, so these plots were computed using this kind of movie. So in blue, it's the cell density. So it's basically the gray level, the opposite of the gray level. And in orange, you have the azimuthal cell velocity. So the velocity of the cell, the component of the velocity perpendicular to the branches. Okay. And what you can see is that, so you have peaks. The blue peaks represent the branches. So you have clearly a very dense and very dilute phase with sharp interface, which tells us that this looks like a phase separation. You can clearly define two phases, a very dense one and a very dilute one. And as we saw in the previous slides, both have completely different macroscopic properties. So all of that tells us that this phenomenon is akin to a phase separation, an active phase separation. And interestingly, if you look at the pink shaded area here, the cells that are close to the branch at the right here, they have a positive azimuthal velocity, which means that they tend to swim toward this branch. And on the contrary, the cells that are closer to the branch at the left tend to swim toward the branch at the left. So you have both a flux of cells to a dense area, and more super importantly, you have non-local interactions. So cells are able to sense their environment over very long distances. Okay, it's not only a cell which is close to the middle, it's a few hundreds of microns away from a branch, so a lot more than its own size, and still it's able to sense that it has to go in this direction. It's attracted by what's happening very far away from itself. And okay, a schematic for the mechanism which leads to this phase separation is here. So what's actually happening is that if you have a small cluster of cells, which can happen randomly initially. So initially you have a small cluster of cells, and so a cell which is located behind, it will perceive here in this schematics more light coming from the left than from the right because of light absorption. So because it's doing negative phototaxis, it will tend to swim toward the right, so in the direction of the already dense cluster. So because of this coupling between light absorption and negative phototaxis, the cells tend to swim toward the dense area, so which amplifies the initial density fluctuations. So you have these feedback loops, which leads to the complete phase separation of the suspension. And importantly, interactions are highly non-local, which is not so common actually in active matter system. Now, if you want to quantify what's happening a bit more, we can use the selected wave number to investigate quantitatively what's happening here. So the wave number is just 2 pi over the wavelength of the instability. And the wavelength of the instability is the distance between branches at the onset of instability, which is well defined, as you can see here in these images. So the first set of experiments that we performed was to increase the cell density. So with same light intensity, we just increase initially the number of cells in the Petri dish. And as you can see, as you increase the cell density, you have more and more branches. Now we can do the same with the light intensity. So it's an independent set of experiments. Same initial cell density, we just change the light intensity. And again, the more light we have, the more branches we have, and it tends to saturate at more or less the same value as in the cell density experiments. And interestingly, I would like you to notice that when the light intensity is very low, these cells are still doing negative phototaxis, so they still go toward the center, so they are still able to sense the light, but there is no destabilization, so there is no branch formation, which is this point here, and this movie here. So here at very, very low light intensity, you just have what we could expect in the first place, which is like a blob of cells at the center. And then the last set of experiments was to increase the viscosity of the medium, but keeping the same light intensity and cell density. And here again, the more viscous the medium is, the more branches we have. And this is these images here. So now we derive the model, the simplest possible model to explain all of these experimental observations, which is the following. So we have 2 equations. So first, cell conservation. So this the rho is the cell density, and this tells us that the cell density can evolve either through diffusion or through advection. And as you can notice, it's a 1D diffusion advection model. It only depends on x, and x here is the azimuthal direction. So what we say here is that we're only interested, the interesting thing here is what's happening not in the radial direction, because this is quite trivial, but in the orthoradial direction, where you have actually the phase separation and the branch formation. And we couple it with this equation here. So we say that the advection term here is the phototactic speed. Is due to phototaxis actually. So we have a phototactic drift, and cells tend to swim toward dense area, which is what we saw in the experiments. And how do we write it? Well, we say that in this 1D system, cells compare the cell density on their left and on their right, and they go toward the denser area. So, and importantly, they compare the density on their sides over a fixed length, which is a kind of an interaction length, delta x. And this finite interaction length encodes the non-local interactions. So they are sensitive to what's happening on their sides over quite a big distance. And this parameter alpha is phototactic sensitivity. So the higher alpha, the higher they will respond to a difference in density on their sides. So now with these 2 equations, we can perform a full analysis of the model. So I won't enter too much into detail, but what you do is you assume a small perturbation in the density, so like kind of the random fluctuation that we expect initially. And then assuming that this is small, you can linearize everything, and then you end up with a relationship between the growth rate of the small perturbation, which is the sigma here in this exponential. So the higher the growth rate, the higher this small sinusoid perturbation will grow, and the wavelength, which is the wave number, which is the k here. And so you do your linear stability analysis, and you end up with this equation for the growth rate of the mode k, so sigma, and you see that it depends on the wavelength and on the interaction length and its sign, which is the most important thing, because if the growth rate is positive, then the perturbation will grow and the system is unstable. But if the growth rate is negative, then the system is stable. You don't have any perturbation are not able to appear. And the sign of the growth rate is controlled by a single parameter here, which we call beta. And beta is actually kind of a Peclet number, so it compares advection over diffusion. And this analysis tells us that if this parameter is that the system will be unstable only if this parameter is big enough. And this is consistent with what we see in experiments. So we have two possible regimes, one where the system is unstable and one where the system is stable. So the stable system, you saw it previously, for instance, at low light intensity, but we also see it, for instance, in this high deficient mutant, which can swim, but which phototactic sensitivity is very low. So it goes toward the center, but without phase separation. And now, if you increase a bit beta, you can reach regions like wavelengths that are unstable. And when the system is unstable, you can compute analytically which. So these equations will tell you the growth rate of each mode, so of each wavelength, and the fastest one will be the one which is selected in this kind of analysis. And so the wavelength that we see in the experiments is the one that grows the fastest. And we have then this analytical formula for it. And we can see that it saturates. So when beta increases further again, it will saturate the selected wavelength, or wave number will saturate at a given value, which only depends on the interaction length, which is also what we saw in the experiments. Like as you increase either the cell density or the light intensity or the medium viscosity, we saw that the wavelength saturates. So this model, which is kind of empirical because the drift equation is empirical, is able to capture the essential physics of what we saw. So an unstable transition between stable and unstable regimes above critical parameter, and then the saturation of the wavelengths as this parameter further increases. So I'm not sure I will go into these details, but it's also possible to map these equations in another famous equation, which is called the porous medium equation. And this tells us that you can understand it as an effective diffusivity of the cell, which can become negative. So, and when the effective diffusivity is negative, this means that the system is unstable. But okay, I won't take too much time here. And okay, now that we have our analytical predictions for the selected wavelengths, we can compare it with experiments. So we need to map our parameters with the experimental parameter, which are the light intensity and the viscosity. We end up with this big formula here. And as you can see, it fits very well with the experiment. So we have some fits parameter, which are the interaction length delta x, and also one other parameter in each set of experiments. But interestingly, these three independent sets of parameters reach the same fit parameter, I mean fit parameter that are consistent, which is a very good sign that our model is actually relevant regarding the physics of the system. And as you can see, what the obtained interaction length delta x is in the order of a few hundreds of microns, which is way larger than the size of a single cell, which is 10 microns, which tells us once again that clearly it's long-range and non-local interactions that are super important here. And this is interesting because I mean, you can show that if you didn't have these long-range non-local interactions, then you wouldn't end up with you could have a phase separate, you couldn't have this finite wavelength selection. And this is something that people usually neglect in active matter studies because it's just easier. Okay, so one of the parameter of our model was this phototactic sensitivity alpha. And interestingly, so with all of this analysis, we are able to say something about alpha, so the phototactic sensitivity in our system, because we fit it. And but this single parameter alpha doesn't say much, but we have to relate it to the parameter that we actually control in the experiment, which is the light intensity. So we assumed that we did a very basic, I mean, classical hypothesis that the phototactic sensitivity of the cell is related to the light intensity with this Hill function, which is a very famous equation for the sensitivity of a receptor, chemical or photoreceptor. So the higher the light, the higher the response is, and the magnitude of the response will depend on this p exponent here, which will tell you something about the cooperativity of the molecules inside the receptor. So it's a very generic law. And k is the response, is the half response. So it's the half, the value of the intensity at which the response is half its maximal value. Okay, and so what I want all of this to say that the relevant physiological parameter here is this p exponent here. And interestingly, so with this fit and this analysis, so we cannot have too much fit parameters, but can see that the fits work best when p is fixed to 0.7. So it's very clear if you look at this plot. With everything equals, you change this p value, and clearly the fit is better when this p, is equal to 0.7. And this p is the important relevant physiological parameter, and very interestingly, it's super conserved in a wide variety of systems that use the same photoreceptor. So we have lots of papers on Chlamydomonas, so on the same algae, or even in transgenic mice that express the same photoreceptor that was taken from the cells. And they do this kind of experiments. They measure the response of the photoreceptor as a function of the light intensity. They use the same Hill equation, and they found the same value for these coefficients. So this is, again, so we just measure the same thing in a very completely different way. So we use this kind of collective phenomenon to extract the same value. But this, again, tells us that these models, the basic model tells us something about the physics of the system. And here, I think I will not take too much time about it, but we also studied what actually sets the interaction lengths of the system. And this is purely geometrical. Okay. And finally, because we have this very good understanding of the system, we are able to plot the phase diagram of the system. So we can plot the analytical prediction for the stable and unstable regime, and it looks like this. and the circles are the experimental value. So it's just another way of showing the experimental results. Okay, and now then we asked ourselves, okay, so here the cells, they all swim toward the center when they are sensed by light. And they are all doing negative phototaxis. So it looks like they want to hide from light. But is it actually working? So are they actually protected in the dense phase? So as you know, I mean, so and measurements of photo damage is something which can be very easily done. It's, I mean, when you have the appropriate scope and light protocol. And so we collaborated with Benjamin Bayeul and Sandrine Jaldon at IPPC to actually measure photo damage in this very dense phase here and investigate if cells are actually protected in the dense phase. So this is the kind of things that Aliénor did in her own custom-made microscope. So this is like the basic protocol of measuring the quantum yield of photosynthesis. So I won't go too much into detail, but you use the fact that fluorescence tells you a lot about what's actually happening about photochemistry, because the more fluorescence you have, the less energy is used for photochemistry. So you have this parameter FV over FM that you can measure in a very precise protocol that tells you about the quantum efficiency of photochemistry at a given moment. And importantly, it decreases with photo damage. So what we did is measure this parameter first in a setup which is tiered. So here we take the whole setup, the cell suspension and the LED, and we steer everything. So cells are submitted to the same amount of light, but they are not able to aggregate in the dense phase because there is no dense phase is possible. It's just like at every moment you just have a homogeneous cell suspension at a fixed cell density. And as you can see here, so when you turn on the LEDs for 20 minutes, and this parameter, the quantum, the FV over FM sharply decreases, which means that you have a lot of photo damage. So the proteins for photosynthesis are not able to use light anymore. So you have a lot of fluorescence, and so the quantum efficiency of photosynthesis sharply decreases, and then you switch off the light and it goes back, it relaxes to its initial value because it's a biological process. You have a repair. So the proteins are repaired and then you go back to the initial value, except when you add this inhibitor lincomycin. So here there is no repair possible, so you stay at the basic value. And this tells us that this is a signature of photo damage, photo inhibition. And then you do the same thing, but without steering the setup. So you let the cells protect themselves into the dense branches and then into the dense blob in the end. And here it's the blue curve, and you can see that there is a big difference in the. So you have a bit of photo damage because this ratio decreases here, but much less photo damage. So we can conclude that indeed you have some collective photoprotection through phase separation. So cells are able to protect themselves collectively through phototaxis. Okay, and just to conclude on this part, finally, we worked with Aliénor, who was then PhD student at the chemistry department of INS, to measure the light intensity field in this kind of weird setup, because it's not a very classic illumination geometry. You have 16 LEDs that are surrounding a Petri dish. So it was not obvious to us what is actually the intensity field here. and we wanted to measure it. So we used the protocol that Aliena developed during her PhD, which is published here in Nature Methods. So I'm sure you know about it. And it allows us, so it's based on, so what we actually measure is the decay in fluorescence of some molecules. And the faster the fluorescence decay, the higher the light intensity is. And this allows us to measure this very beautiful, I think, image here. So this is just the light intensity field when you don't have cells. So it's just a petri dish which is filled with water and with these fluorescence molecules. And the red parts are red means high intensity and blue low intensity. And curiously, you have more light at the center than at the edges. So we didn't expect this at first because, light is supposed to decrease as the inverse of the square of the distance. So we didn't expect that at the center, which is the farther away from the light, you have more light. But this is actually due to the fact that you have a lensing effect from the petri dish. And you can also model everything. This was done also in this paper. And okay, I like a lot this image because you can clearly see the refraction of each individual LED. So it's super clean. And interestingly, now when you add cells, the pattern changes. So now you add cells. So here they are dead. You don't have the pattern. It's just like similar to what you have at t equals 0. Just have a dense suspensions of cells without the pattern. And here, you have more light actually at the edges than at the center. And this is due to light absorption by the cell. So here, when you go toward the center, you don't have you have much less light because everything is absorbed by the cells at the edges. And as you can see also, we cannot see anymore the sharp rays of light. And this is due to light scattering by the algae. Okay. And this is also interesting for us because it tells us that here the gradient of light intensity switches signs. So between the low cell density and high cell density experiments, the gradients of light intensity changes. But as we saw in the experiments, the cells always end up at the center. So this tells us that in this particular geometry here, cells don't follow the light direction. Cells don't follow the light intensity gradients, so they just but the light direction. And this is actually kind of an open question because it seems that in other elimination geometry, you have to take into account the light intensity gradient and not the light direction. And people are trying to understand at the single cell level what could explain these different mechanisms. Okay, so this is the first part of the story on phase separation in this phototactic microalgae. And actually, at some point, so we started, we saw everything, we started with this circular geometry, but this was actually by chance. And at some point we thought, okay, but maybe we should do it in a linear geometry because everything will be much easier, notably to compare it to the model. So we tried to do this same experiment, but in a linear geometry. So instead of having 2 circles, you have 2 lines. So one array of cells, and then just a rectangular cube with the same cell suspension. But actually, this is what's happening if you do the same in a linear geometry. So, okay, you have the phase separation and the branches, but very quickly, you have secondary instability that maybe you can see here, which is the fact that you have a destabilization of these branches into droplets, which looks like the Rayleigh plateau instability, when you open when you just look at a thread of water, it will also break up into droplets. So it looks the same. But of course, not the same, because here, as you will see, it's an active process that is due to hydrodynamic interactions between the cells. And so this second part is a project that we did in collaboration with a team of theoreticians at Cambridge, so Marco Vona and Eric Loga, and Takuji Ishikawa in Japan did all of the simulations. Okay, so now it's the same cell, Chlamydomonas reinhardtii, but we change a bit the perspective, because now we're not going to investigate phototaxis, we are going to use phototaxis to have them interact in a very precise way. So here, now we are going to use the fact that each cell is swimming. So it pulls on the fluid, which it's two flagella here that are located in the front. And so it's actually a very well-studied type of microswimmer, which is called a puller, because it pulls on the fluid. And if you look at the flow field it creates, it looks like this. So here the cell is swimming toward the right. And so it's pulling on the fluid in the swimming direction. And by mass conservation, it's pushing on the fluid on the opposite direction, in the perpendicular direction. So now, if you put a lot of these cells swimming one behind each other, you will expect them, because they are pullers, to pull on their neighbors. And so if they swim one behind each other, you will expect them to cluster, which was predicted in simulations here. And on the contrary, if you have them swimming side by side, now they will push on their neighbors. And so you expect to have some buckling, which was also seen in simulations here and here. And so what we did in this project is investigate it experimentally for the first time. So the setup is once again very simple. It's a cube, like a few centimeter long cube with a suspension of swimming algae. And on its side, so this is the top view, on its side you have two linear array of LEDs. So in the dark it's homogeneous. Then first we want to create a cell jet. So we focus them at the center using light and negative phototaxis. So they form this very thin band at the center. And then, okay, some schematics is missing, I'm sorry. And then at t equals 0, you switch on off the side side lights, and you switch on. Sorry, you switch off the side lights, and you switch on a light which is just parallel to the line, the cell line. And this looks like this. So they will all start to swim one behind each other, and actually, as expected, very soon the cell jet breaks up into droplets, and then you have other things that happen. And this clustering instability can also be recapitulated in simulations. So in these simulations, the only ingredient is hydrodynamic interactions between the cells. So clearly, hydrodynamic interactions are enough to explain this clustering, and even apparently what's happening afterwards. So the stretching of the droplets and then even the merging. So this is the microscopic view. So here I'm following the drops as they move in order to see their long-term evolution. And as you can see, you have a lot of complex things that happen. And in the end, you have this very dynamic V shape with big, large-scale recirculations here. Okay, now we play the same game. So we initially did the same system. And then here, simply, we just let them swim toward the center. So perpendicular to the line, we just increase at t equals 0. We just increase the light intensity on the sides. So here, in this geometry, each cell will swim perpendicular to the line. So you will have lots of cells swimming side by side. So instead of pulling on their neighbors, they will push on their neighbors. And we see what was also expected, which means here buckling in this geometry. So now if the micro swimmers are aligned perpendicular to the band, you have this buckling instability. And we also see it in simulations. So now it's simulations of micro swimmers that are swimming side by side, so perpendicular to the line. And you can also see this buckling happen. Okay, maybe I will go a bit faster here, but then Marco Vona and Eric Loga did this amazing job of writing the full 3D analytical model of the system. So the hypotheses here are a dilute jet of micro swimmers and micro swimmers whose swimming direction is clumped. So they have a fixed direction, either parallel to the cell jet. And under these assumptions, you can also do a linear stability analysis of a boundary perturbation. So initially you have like just a cylindrical cell jet, and you slightly deform it. So the cell density is constant, but you just change the shape of the jet. And the model tells you that this boundary perturbation can be unstable. And depending on type of swimmer, you can have either the clustering instability or the buckling instability. And so these models recapitulate both the purling and buckling instability, but it also gives us an analytical prediction for the growth rate of the mode, so the speed at which the instability will appear. And it also gives us analytically the wavelength which is selected. And as you can see, the wavelengths only depend on the initial width of the jet. And now we can compare it with experiments. So this is what is plotted here. So here it's one example of the amplitude of the instability for the drop. So, and as you can see, so first we have an exponential rise of the amplitude of the instability, and then it saturates. And if you compute and the growth rate, which is the slope of this initial rise, increases with the light intensity. And if you compute the wavelength at the end of the exponential rise, and you compare it to the initial cell width, jet width, you get these points, which are consistent with the model and simulations. And then we can do the same with the zigzag instability. So you compute the amplitude of the instability, which is just basically the amplitude of the sinusoidal shape of the jet. Over time, you also have this exponential rise, then saturation. And here, the growth rate has this complex relationship with light intensity. And the wavelengths also seem to be consistent with the model. So you have to keep in mind that the model seems to work very well. Also, the simulations tells us that pure hydrodynamics interactions are enough to explain everything which is happening here. But we still have some discrepancies between model, simulations, and experiments. And the main one is that the model and the simulations assume that the jet is dilute. So that the cells are quite far away from each other, and so they only interact through their far field. And of course, this is not actually exactly the case in the experiment. So this explains slight discrepancy that you have between experiments and model. Another thing that we can investigate is the flow field around the jet. So in order to do this, I did some PAV experiments. So here it's dark field, so the white part are cells plus some passive beads that I added. And the motion of the beads, which are just passively advected by the flow, will tell us about the motion of the fluid. So this kind of movie allows us to plot this kind of map of the flow field around here, the droplet instability. So as you can see, you have big recirculation like this around the droplets. And this is the prediction from the model. And we can also compare quantitatively the decay of the flow field around the jet. So of course, when you go far away from the jets, the flow field goes to 0 on average. And what we see in experiments is also consistent with the model. Then you can do the same thing with the zigzag. You add beads. You can see big macroscopic recirculations of the beads, which tells us that you have big vortices in the fluid around the jet. So as you can see, fluids have fluid motion from the dent to the peaks of the zigzag. And we can also compare it with the model, and it's also very consistent. Okay, and now to end up this story, I just wanted to show you something which I think is very nice. So the previous experiments, in these PIV experiments, you follow beads, not the algae. So here you just follow the fluid, the motion of the fluid. But we can also follow the motion of the algae. So here I did PIV on the motion, and so what you end up with is just the cell displacement field. So this is in the drop frame, and as you can see, so here this droplet is moving toward the top of the image. This is also a top view, and it's behaving a bit like a big puller. So it pulls on the fluid in the direction where it moves, and it pushes on the fluid in the opposite direction. You also have these vortices around it. And this is very similar, at least qualitatively, to what your model predicts. So this is the prediction of the model. You pull on the fluid in the direction where you go, you push on the fluid on the opposite direction. And Marco Vona, which is a PhD student of Eric, did this full analytical computation. So he showed analytically that the circular ellipse, only because of this flow field here that they create, they will elongate into ellipse. So you can understand it here in this model, in this picture, because they are pulled on the side and pushed here on this direction. So you can get that the ellipse will stretch, and they elongate in a self-similar dynamics. So the evolution of the shape is independent of the initial shape. And also, funnily enough, each big cluster of cells will then behave like one single cell. So he showed this analytically that under some specific conditions, the flow field created by a cluster is just and the number of cells times the flow field created by one cell. So you end up with droplets and they each behave like a single cell. So they will each attract each other. So to conclude, you start with a vertical line of microswimmers that are pullers. So because of these hydrodynamic interactions, they tend to cluster and form droplets. But then, also because of hydrodynamic interactions, the droplets stretch into ellipses. And so you end up with a horizontal line of puller, like here. But a horizontal line of pullers, it's actually what we have in the zigzag instability. So at first, it's a vertical line of pullers that are pulling on their neighbors, and you end up with a horizontal line of pullers that are pushing on their neighbors. And so actually what you end up with is the zigzag instability. And this is exactly what we see in the experiments. So this is a clean example of the long-term dynamics of a cell cluster. And clearly, it's exactly the same of the zigzag. So the cell displacement field is the same. The angle between the two parts of the zigzag is the same. And we only have one wavelength. It destabilizes into zigzag as soon as you have, as it's long enough to have one wavelength. So this is what you end up with, this kind of structure. So I think it makes a nice story. And the only player here is hydrodynamic interactions. You don't need anything else. And I just wanted to conclude by saying that this was a story about micro swimmers, but the kind of pattern that we show is actually much more generic than this. And because the only ingredients that you need in model and simulation is just the structure of the flow field around each cell, if you take a completely different system with completely different particles, but they have the same flow field around, but they generate the same flow field, then you should end up with the same structure. And this is actually seen in a lot of different systems. So, for instance, if you take colloids and you put them in alternating electric field or magnetic particles in an oscillating magnetic field, you can show that the flow the flow they create is the same. So pull in one direction, you push in the other direction, and so you end up with the same structure. And so this tells us also that the model that we derived could be used to understand all of these very different phenomena. Okay, and with this, I would like to thank everyone. So the team where I did my PhD, and Raphaël and Nicolas, and of course, people at Cambridge, Takuji, Benjamin Sandrine, and of course, Aliénor, with whom it was a pleasure to collaborate. And thanks a lot for your attention.
Thank you very much, Isabelle. So maybe we have time for a few questions. Are there any questions in the audience? Yes, Aliénor.
Yes, thanks a lot for your talk, Isabelle. I just had a question about the active PIV versus passive PIV.
Yes.
So are you modeling different flows, or do you, like, is the conclusion that algae are just passively following the flow? I didn't understand if there was a difference between what you...
So actually both things are consistent. So cells are creating the flow, but they're also advected by the flow. So here what you can see is that the cells, here the cell displacement field just tells you that the circular droplets will tend to elongate because cells tend to go into this direction here on the sides.
Okay.
And this can be explained also by the flow field. But this is not plotted in the same reference frame, so maybe it's not super easy to see here.
Okay, and thanks. And I just had one other question. It's at the first part of the talk. It's can you explain again, when you have two branches that start merging, what's the explanation? Like I understood how algae migrate towards the branches, but do you also explain that this is just because you need to at some point reach the wavelength of the system? But can you explain the dynamic?
Yeah, it's a very good question. So actually, when you have two branches that are, so I think you need two things for branches to merge. So they need to be close to each other, so that cell in one branch will be sensitive to you need the cells to be in the shadow of the neighboring branch. So they need to be close enough to each other. And also you need the density in the branch to not be super high, because if the density is super high, then the cells inside, they don't see anything. So you need a branch which is not yet extremely dense and close enough, and then it will be attracted. And we can see it like, for instance, if we use a super strong blue light, and then you have lots of very thin but very, very dense branches, and it seems that they are more stable, like they merge less, and probably it's because they are so dense that they cannot really feel what's happening on their side.
Okay. Well, thanks a lot. I will have other question. I will write to you.
Sure.
And I was surprised to see that even at low light intensity, the cells have negative phototaxis, but I would expect there is a range of luminosity where you would have positive phototaxis. So how do you explain that?
Yeah, indeed. So this is due to the fact that first, this strain is super negatively phototactic. And this is also due to the way we acclimate the cell. So here the cells, before each experiment, we put them 20 minutes in the dark. And under these conditions, they this strain almost only do negative phototaxis. And indeed, you have papers where they see a switch between positive and negative phototaxis, but you have a different acclimation protocol. So I think they put it like 1 hour under red light, and this induces physiological changes. But under our condition, we only have negative phototaxis. Okay,
thank you. And also, I was curious, how do you measure the velocity in the study you showed, where you showed the azimuthal velocity? How do you measure? Do you track each cell? Or?
Yeah, exactly. It was just single cell tracking. So when it's possible, so when it's not too dense, and and you can do this.
Okay.
And on that topic, have you managed to have algae of different fluorescence color that you can easily spot in a mix? Or
no, this is an idea I think we had at the very start of my PhD, but we never managed to find this strain. So yeah, so what we wanted to do is follow what's happening inside the dense phase, and it's super difficult because it's so dense. So we wanted to add a few fluorescent cells, but then it's difficult because you have to find a wavelength which doesn't interfere with photosynthesis and which doesn't interfere with phototaxis, and this is quite difficult to find. So no, we didn't manage to do that.
Okay.
And if you add positive phototaxis, do you have predictions on what patterns you would have, or
in the branching experiments? Yes. So actually, you can show that if you have positive phototaxis instead of negative phototaxis, so somewhere in one equation you have a plus instead of a minus, and then the growth rate will always be negative. So this instability is only possible with negative phototaxis.
Okay.
Yeah. Because you need this feedback loop, and if you have positive phototaxis, you don't have it actually. You have only two things that go in the same direction. So diffusion and positive phototaxis both tends to homogenize the suspension.
Yes. And also, you add a D0 parameter in your formula?
Yes.
And you don't feed this?
Sorry, I don't what?
You don't estimate this parameter?
Yes. So we computed, we measured it actually in a separate experiment. So D0 is the diffusion coefficient of the algae, and we measured it. So you just concentrate some cells, and then you just switch the light off. You just see how they diffuse. So this is very easily done. But the thing is, it's diffusion in the dark. So it's for sure it's underestimate the actual diffusion.
Okay. Thank you very much. I think I see no hands raised.
Yeah, just have one last question. Is that, did you try to do, this is transmission imaging in the setup?
Which setup? the branches?
I mean, both like for imaging, you use transmission, so light on one side and image on the other side.
Yeah.
And have you tried to image the fluorescence to see if we could like some sort of find similar conclusions to what you found with the Benjamin and Sandrine experiment on fluorescence, like the fluorescence distribution in the sample through time versus some simulated fluorescence evolution?
So I think, at first we wanted to have a map of photo damage, because what I showed was just the bulk average photo damage. So we wanted to do it with a spatial resolution, but it ended up being super difficult because, it's extremely dense. So only the top layers see the light. I mean, from the see the excitation light. And also, when you want to follow this protocol, you have to wait a bit, and they move too fast for this. So we didn't do this.
Okay. Well, thanks a lot. And it's super elegant how your models fit the data.
Thanks.
Okay. Yes, thank you very much indeed, Isabelle. And see you all next week, where the topic will be human-robot interaction, I think. So goodbye.
Bye.
All right.
Ciao, thanks a lot. It was super nice. Thanks.
Thanks.